All right. Hi, everyone. So this is the exam one review for Bio 285, and we're going to go ahead and get started. So before I discuss material, I figured I would put some advice for how to go about this class. First thing I'm going to mention is your exam is tomorrow. Hopefully that's not a shock to any of you here, assuming you guys knew that. Sorry. And so thing I wanted to mention is that today's slides do not cover every single thing that you should know for your exam. You might be asking why. It's because if we tried to cover everything that would be for your exam, we'd possibly be here until like 1 a.m. trying to explain every single thing. But I don't think anybody wants that. So we just explained a vast majority of content that you will find on your exam, but it's in your best interest to make sure that you review things that we don't cover in this review. You can watch previous review videos on YouTube. We made sure to post those every week. You guys have like your class notes. We have a TA that sits in lecture. Emily, she writes notes during class. So if you were to miss class, if you were sick, you have access to those notes on Moodle, and just any other classroom resources you can use to study. Highly recommend that. But I'm just gonna go through just more tips for your exam. Make sure that when you're doing your exam, make sure that when you're doing your exam, that you read the question prompts carefully and you take time to answer the questions. A good thing about your exam is that Professor Francis is not one of those professors that after 1 hour or something or 2 hours, she's gonna come through and yank all your exams and collect it really fast. She's actually very nice that she's given you enough time to do your exam. I speak from personal experience. When I was a student in this class, I was always the last one to leave the exam room. Why? Because you just get the pleasure of being able to have all the time you need to do it. So just take your time, read through things carefully, ask questions when possible. And because these questions are mostly problem solving type things, so making sure not to just rush through it in an hour. Yeah, some other things I mentioned is just being able to use the resources that we provide to you, both the TAs and Laura, but don't rely too heavily on these. Use your own notes, use quizzes for review of more than just multiple choice, and try to think about how questions can be turned into other questions and create your own questions when studying. More advice, Laura told me, Professor Francis, but she doesn't mind your title, says definitely to engage in active studying. When I was a sophomore taking this class, I learned all about what that means. And active studying just means that you're engaging with the material in a productive way. So being able to do practice problems, being able to work through your problem set with other people actively, thinking of what would happen if I mutate X, Y, or Z, things of that nature, drawing out pathways by hand. Well, we haven't done pathways yet, but drawing out like certain things by hand, because it's proven that when you write stuff down, you remember it better. Hence why your cheat sheet is handwritten only, not typed only. And yeah, and also starting the problem set early, because some people like to wait until Saturday or Sunday night, but don't do that. Don't stress yourselves out. Try to start it early, because the sooner you start it, the better. And so that's just the difference between active studying, passive studying. Passive studying would just be like sitting there, rewatching the lecture video, reading a book, just not really practicing it physically yourself. So yeah, those are just more advice things. And like I said before, your exam's tomorrow. I think, did Professor Francis give you guys like a place where you sign up to do your exam? Like each person has like a different time according to their schedule. Can people give me a thumbs up if what I'm saying makes sense? Or thank you. But yeah, so the exam is tomorrow, but depending on what time slot you signed up for, the time is different. More things on advice, I'm just going to quickly say, utilize things like iClicker questions, practice problem sets, quiz questions, and try to come up with your own questions. And another thing I'll mention is do not waste time memorizing like big diagrams, things like trip operon on the amino acid sheet that you got week 1 and nuclear export import. Those diagrams will be provided to you on the exam. It is the last page on your exam. You get a sheet that has like the picture of all those things on there. So don't try and memorize those. And sorry if you put it on your cheat sheet already, you might have to erase and put something else, but you do get those on the exam. So that's nice. You don't have to memorize those. And like I said, the cheat sheet's double-sided, correct? Double-sided sheet of paper, handwritten, can't be typed. And just, I recommend not putting every single thing on there because I've had a cheat sheet before in other classes. And like I used to just write every single thing on there. And you end up being more stressed because you're spending time trying to locate what you wrote in size, like -2 font, trying to figure out where is this thing I wrote in what corner. And that causes you more stress than you actually kind of like engaging with the material. So unless you have like a magnifying glass or something tomorrow, I'd recommend not putting every single thing on there and just putting things that you think would be useful to put there. Does anyone have any questions on anything I said so far? No? And lastly, just want you guys to relax, take a deep breath, and that you guys have the tools necessary to be able to do this exam. And we as the TAs and Laura have faith in you that you're going to do well. I think sometimes positive affirmations are good in times like this, but you guys all seem stressed the way you're all looking at me. So, okay. Yeah, so we're going to start with like actual content now. So
all right, guys. So week 1, we learned about amino acid and levels of protein structure. So that's what we'll go through first here, okay? So, you see my, yeah. All right, so just like the basic structure of every amino acid is we have a central carbon with a hydrogen. We have the R group, which differentiates all 20 amino acids. We have the amino group, and we have the carboxyl group, right? And just a side note is amino acids are protonated in low pHs and deprotonated in high pHs, right? Now for labeling amino acids, we go from left to right or N terminus to C terminus. So this would be amino acid 1, 2, 3, 4, and going down the line. And know that R groups are arranged by chemistry and size. So that's to do with stereochemistry where we don't want 2 huge R groups next to each other. All right, so the first level of protein structure is primary structure. And this is the overall general sequence of amino acids. And this is held together by a covalent bond called a peptide bond. So peptide bonds are very strong. They actually mimic double bonds due to resonance. So they're very hard to move around their axis. And just know it requires a lot of energy to break them. And to do this, we need enzymes. All right, so our secondary structure is going to be the alpha helices and beta pleated sheets. And these are between backbone atoms. So the hydrogen, and here you can see right here, the hydrogen on the amino group is going to be forming a hydrogen bond with the oxygen on the carboxyl group of the backbone atoms, right? So this is due to electronegativity differences. So the nitrogen has a higher electronegativity than hydrogen, so it pulls electrons towards it, giving hydrogen a slight positive charge, and oxygen already has a slight negative charge. So we get a dipole-dipole interaction, and hydrogen bonding is a type of dipole-dipole interaction. Remember, these are non-covalent interactions. Also know that alpha helices, the R groups are on the outside, and on beta pleated sheets, they alternate up and down.
I forgot to say earlier, but if at any point, if you guys have questions, please raise your hand or just feel free to just say, excuse me, can you explain this again? All that stuff.
Is there any questions right now? We're good. All right. All right, so the third level of structure is tertiary structure. This is the overall like 3D shape of the polypeptide, right? So just know that a polypeptide is a subunit. We'll get into that a little bit later. So the polypeptide folds, this can be done through hydrophobic interactions. also known as van der Waal interactions, ionic bonds, hydrogen bonds, disulfide bridges. I'd also make sure you know that disulfide bridges are the only covalent bond you're going to see in tertiary or quaternary structure. So that's between the cysteine amino acids. So if Laura says, there's one disulfide bond, we know that there's two cysteine amino acids that are taking part in that. Any questions so far? Good. All right, and then quaternary structure is just a bunch of polypeptides that come together and form the same ionic bonds, van der Waal interactions, disulfide bonds, hydrogen bonds. It's just two or more polypeptides that come together. All right, so more on tertiary and quaternary structure. So tertiary structure, a single polypeptide chain, and the way it folds in 3D. Quaternary structure, multiple polypeptide chains folded into a 3D shape, stabilized by covalent and non-covalent R group interactions, along with polypeptides, hydrogen bonds, electrostatic interactions, van der Waals. All right, so that's just a quick diagram. So we go from primary structure, right, into secondary structure, which is the alpha helice, and then we go into tertiary structure, which is the overall fold of the polypeptide, and then quaternary structures, and we take a bunch of polypeptides and put them together to form a protein. All right, so domain versus subunit. So domains, I guess we'll start with subunits, right? So let's take a look at hemoglobin here, right? So a subunit in protein in quaternary structure are the individual polypeptides that makes up the protein, right? So for example, yellow is one subunit, orange is another one, or actually these are, yellow is one, orange another one, blue is another one, green is another one. So those are all subunits that come together to make a polypeptide, right? Then we have a domain. So a domain is a section of a protein that folds independently from the rest. So it usually carries out a specific function, and domains are evolutionary conserved, right? So they can be, a binding pocket for ATP in, a kinase, for example. So it says here, within tertiary structure, there are independently folded sections of the polypeptide. They are still connected by other parts of tertiary structure through the polypeptide connections, but the bonds used to form these sections fold and kept within the R groups. So same stuff we did. Sorry.
I wanted to mention, I see people writing down stuff, but you guys are welcome to take notes, but I'm going to make sure this is emailed at the end of the review session. So you guys will have access to everything. So don't panic if this slide changes anything. Get down. It will be all sent.
So any questions thus far? So think about it as in tertiary structure, we have polypeptides, right? Each polypeptide can also be called a subunit, okay? Now a domain, so imagine now we have an overall protein, right? Whether that's made of one polypeptides, 2, 3, 4, whatever. So a domain is like how that part of the protein folds. So it's a group of amino acids, which are in a subunit that fold in a certain way for a certain interaction, right? So like you said, ATP binding domain in a kinase. So imagine it's like this part of the protein wants to fold by itself in this section, if that makes sense. I know it's a hard concept to understand.
So your question was difference between domain subunit. So subunits only exist in quaternary structure. So when you have two or more subunits, that's defined as quaternary structure. Quaternary structure can also have domains inside of the polypeptides, the individualistic parts of subunits, which is where it can get confusing. But tertiary structure is just one polypeptide, a single polypeptide, so a single protein, and that protein can have a domain in it, and the domain can have like a special job. I'm trying to think of an example other than ATP binding domain, but like if you had like a Hox gene domain, like in a monkey, and a Hox gene domain in a human, like they both form the same thing. So that's what we mean when we say like a domain's evolutionarily conserved, will carry out the same function between animals. And yeah, so I don't know if that helps, but subunits are only associated with quaternary structure. It would be wrong to say that there's a subunit tertiary structure.
Oh, sorry, I was just talking about.
Do you want to add anything? But yeah, did that make a bit more sense? Any other questions? We got a question right there. Hmm, There's just. So as I don't want to confuse you guys more, but just associate subunits with quaternary structure, and that like, sorry, need to go back to this one. And that like subunits is just when you have like two or more kinds of polypeptides within a protein. So I don't know if you guys know about like hemoglobin. Hemoglobin is a protein that moves around in your bloodstream, carries around oxygen. And hemoglobin has subunits in it, like different heme groups that are different colors. And so like subunits are like small polypeptides, like small little pieces of proteins that make up a mega protein. Like that's the way I kind of look at it. There's an analogy here. Sorry, I just wanted to show it so that, no, I don't want to do that. I'm not doing it. I wanted to just see if I can show an analogy of that. Sorry.
No.
Okay, so I can actually show that at the end of the review. I'm gonna show that at the end of the review picture. But yeah, do you want to explain the analogy?
Oh yeah.
Okay.
Can you guys all hear me? The way like I don't know if any of you here for like the first week's review, we went over this. The way we like kind of explained it was think of like if you have a train, you could have a train that's either made of like multiple like cargo like subunits or just like the one little one. So if you think about like a protein can either be defined as like a molecule with just a single polypeptide chain, right? But if you also could have like proteins that have like multiple different polypeptide chains like joined together. So in that case, like each of those chains is considered a subunit. And on those subunits, you would have regions of like important like DNA segments that would be considered like domains that code for like certain like genes or proteins. So does that make sense? Like, if you think about how you think about how like a train has like defined parts and like each of those parts has like like a window, wheels, stuff like that. Like those distinct parts you can think of as like domains on a polypeptide. Does that kind of like your
Okay, so I'm really frustrated at myself because I had the slide for this, but I didn't add it in here. But another example that might help to make it make sense is like thinking about like people. So if like let's say that I represent tertiary structure, like me as a person, it's kind of weird to think about, but just let's imagine I'm a protein, but like I'm also like a person as well. So as a tertiary structure, a person can have like individual parts. So like I have arms, I have legs, and like my arms can do different things that my legs can do. So that's like could be an example of like domain kind of, because they're both parts of me, but they perform different functions. So like that's like an idea behind domains. And then how I would link that to quaternary structure would be like if I went and held hands with Zayed, and we would be like two polypeptides, so like me, Zayed, and David held hands, like that would be two or more polypeptides, which would be subunits. So we're all proteins, so we'd be part of like a mega protein complex. So it would be like I'm a protein and they're proteins, but by linking arms and connecting together, it would kind of look like the thing up there on the right, because we're all like different, multiple polypeptides, which represents subunits. But each one of us still have our like individually functional parts, so like arms and legs, and each perform different functions. Does that make a bit more sense, or does that lose people? No, can you give me like a thumbs up? Thank you. Question?
Yeah, so you know how it says that it has to be two or more different kinds of units? What if you have two like distinct units with other ones? So these two count as two or as one?
We have like two subunits who are identical.
Yeah, and there's like other types too. But do those count as one subunit or as two different?
I feel like they should be considered like two different subunits because they be joined by like covalent bonds, you know. So if there's like a break,
And most of the time, like if you see quaternary structure, if you were to see like a picture of that, it would be outlined in the picture that they have like different subunits with like the different color schemes. But yeah, with the domain, like it might be, you might not always be able to visually see that. So it might be drawn next to a subunit that this one contains like an ATP kinase domain. This one contains like a different domain. Like it would have to be drawn into the picture. You would be able to see that. But with subunits, you can visually see like different kinds. Like I see 4 kinds there. But if there were 2 yellow ones, and if there were 2 yellow ones, 1 green, 1 blue, then that would be like 3 different subunits. So does that make sense? Okay. Any other questions on that? My computer always does.
Oh, that's your computer.
Yeah, it's just on my computer thing. We can switch.
can you guys all like hear me, like especially in the back? All right, so like now we'll go over our group, the different kinds of R group interactions, bonds, and other stuff on amino acids. So the first 2 kind of like subgroups of types of R groups in amino acids are polar versus non-polar. And pretty much all you need to know is that if you have like amino acids that are polar, what does that mean? That means that their R group ends at atoms that have an uneven share of electrons. So a popular example of that would be like how there could be like an oxygen and hydrogen paired on top. Oxygen, as you know, has a very like high electronegativity, so it's really good at attracting electrons, versus hydrogen has very low electronegativity, so it's really so it's easier for it to like let go of its electrons. And because of that, you could have molecules like such as here, like water, that has a strong uneven charge. So where like the electronegative atom would be very negatively charged and the hydrogens would be very positively charged. And you could see examples of that in oxygen, hydrogen bonds, fluoride, hydrogen, nitrogen, or sulfur. So if you ever see like amino acid R groups ending in any of those atoms, then you know that there's a good chance that it's a polar R group. And on the flip side of that, you have non-polar R groups, and these are compounds where the electrons are evenly distributed or they have a symmetrical shape. And if you think about like in this example here, you have carbon and then 4 chlorine atoms, and all those have very close or similar electronegativity. So they're going to be sharing their electrons pretty evenly throughout, and that's going to result in having no overall like distinct partial charges. And then you have the different kinds of R group interactions. And like we said earlier, you'll have this chart, but just so you don't have to memorize like which amino acid is polar, which is non-polar, but just know if you can explain like why an R group would be non-polar versus like why it would be like polar charged or negatively charged. And so for polar charge R groups, those are going to be the ones that are going to make ionic bonds, and they're going to have electrostatic interactions with each other. And as you can see on the table, they're going to have either positive charge or a negative charge. And then you have the polar uncharged R groups. These are going to typically be making hydrogen bonds and they're going to be hydrophilic. So they're going to have a lot of either carbon oxygen, carbon nitrogen, OH or NH covalent bonds. And the other thing with this, like we said before, they're going to have a difference in electronegativity. So one side is going to be more electronegative or negatively charged than the other. And then you have lastly the nonpolar R groups, and these are going to be hydrophobic. So they're typically going to have a lot of carbon-carbon or carbon-hydrogen bonds. Like you can see in the section that says nonpolar, all of them have either just a hydrogen, a methyl, or a benzene ring. So they're going to be pretty like big, bulky R groups relative to other ones. And they're kind of experiencing van der Waal interactions, or also called London dispersion forces. And since they have similar electronegativity between, say, the carbon and hydrogen bonds, they're not going to have any charge or partial charge on them. Are there any questions for the different kinds, or are they? And then our group interaction in tertiary and quaternary structure. Pretty much. Oh, so pretty much in the tertiary and quaternary structure, you're going to have your polar charged R groups that are going to be making ionic bonds with each other. For example, amino acids such as lysine and aspartic acid. And they're going to be experiencing electrostatic interactions. So pretty much any amino acids that has a polar charged or uncharged R groups, such as like lysine or serine. And then you have polar uncharged R groups are making hydrogen bonds with other R groups on the backbone. And this is only true for the tertiary and quaternary structures. Know that the polar uncharged R groups in the secondary structure will not make any like hydrogen bonds. And then lastly, you have the nonpolar R groups, like we said earlier. They're going to be making van der Waal or London dispersion forces. And these are like the lowest energy bonds, so they're the easiest to break. And they're known for their hydrophobic effect, where they tend to cluster or clump together, especially in hydrophilic solution or solvent. And that's what you see as like the hydrophobic effect. And then lastly, you have disulfide bonds or disulfide bridges. And these are covalent bonds, and they only occur between two cysteine molecules because cysteine is the only amino acid with sulfur hydrogen R groups. And they are typically used in the tertiary and quaternary structure because they are covalent bonds. So they are like typically much stronger than other types of interactions. And that'd be important for a protein in the tertiary and quaternary structure for stability. And it's also the only covalent bond in the tertiary and quaternary structure. So all the bonds that we talked about on the previous slide, they're all non-covalent. Yep, and so a quick summary you have of protein or R group interactions. You have either covalent bonds, which can be peptide bonds in the primary structure, or you have disulfide bridges / bonds, which are in the tertiary quaternary structure. And these covalent bonds are much stronger, so they add much more stability and rigidity to the overall protein structure. And then you have, compared to that, non-covalent bonds, and these are going to be all the other kinds, such as your ionic bonds, which are between polar charged amino acids, hydrogen bonds between the uncharged amino acids, and then your van der Waal interactions, which are between non-polar. Are there any questions on that? So how does a protein fold? Like we said earlier, the hydrophobic effect plays a big part in that. So the nonpolar amino acids, because they have interactions between each other, they're going to tend to, the carbon hydrogen or carbon oxygen bonds are going to tend to cluster together, and that, and especially in the cell, which is a hydrophilic environment, that's going to result in proteins spontaneously folding to where the hydrophobic parts are going to be towards the inside, and that's going to expose the hydrophilic R groups towards the exterior. Yeah, so yeah. Any questions on that? Oh, Oh, like what like defines like a nonpolar or hydrophobic? Oh yeah, I can. Yeah, yeah. So like relative, so like when you look at like a nonpolar amino acid, and you see how like virtually all of them have like either a lot of carbons at the end or a lot of like benzene rings, which are just like, like 6 carbon atoms. Those are going to be nonpolar because they don't have uh because carbon has very like low electronegativity, so it's not gonna like take any electrons away from like the hydrogen's bond to. So it's gonna have like an even distribution, and because of that, it's not gonna be attracted to like say like water, which is very like polar because in water you have like one oxygen that like hoards all of the electrons versus like the others. So because of that, it's not gonna have any interactions, and like those carbons are gonna clump with themselves because they have such similar like electronegativities, like if that makes sense. Yeah, yeah.
She wouldn't make me do that for this exam. Okay. She won't. She never made me do that when I took this class. But yeah, you don't have to memorize like because the amino acids are grouped into like the 4 groups there. But some of them in those groups, like for example, like for like cysteine, you could argue that even though it's non-polar, like the SH group makes it slightly have like what's the word I'm looking for? High hydrophilic tendencies or whatnot. But like for yeah, you're not gonna be asked to rank like the nonpolar from the nonpolar groups from the most to least hydrophilic, if that's your question. But what defines all those nonpolar amino acids and makes them nonpolar is the R groups. And the reason why is because the R groups have like the hydrocarbons and the CH bonds. And I don't know, I feel like most of you have taken Orgo or enrolled in Orgo, but you don't need to go in extreme detail about this is a benzene ring, et cetera, et cetera. But just being able to identify that these CH bonds, the hydrocarbons that are featured in the R groups of the nonpolar amino acids, is what gives the amino acid its character. And that's why things in there like leucine and things in there like leucine, alanine, like this is why these ones are classified as nonpolar, because these CH groups are they're similar in electronegativity to carbon and hydrogen, so they're not going to um, they're not they're going to be nonpolar and they're going to make van der Waal bonds. So those are the types of questions that you would probably see, like being able to compare amino acids that are like obviously different. So things like comparing leucine and tyrosine and like figuring out like why would they be classified as different? Like what is it about their chemistry or about the size of the R groups? Like what is it about the R groups that makes these two different amino acid categories? Or possibly where could you find amino acids based on their chemistry? And that ties back to what Syed was saying just about the way a protein folds. Hydrophobic proteins, hydrophobic, like having a phobia, phobic means like water fearing. And because water is polar in the cell's aqueous environment, those non-polar amino acids are going to want to clump up in the center of a protein. Whereas your polar hydrophilic amino acids, water loving amino acids, they're going to want to associate on the outside, the outer surface of the protein. So those would be the types of things that I think she would want me to look for. Does that make sense?
Uh, isn't.
So in a protein wrapped up like that, like in its circular way, I do not believe she would ask you that. But when we get to talking about protein localization, like when the ribosome translates a protein and synthesizes a protein, it's always made with the N terminus first and then the C terminus last. So we will get to that. But in the protein as a ball, I couldn't tell you. And if I couldn't tell you, then I don't think she would ask. Any more questions on this stuff here? No? I like the questions though, they're good questions.
And I'll do protein modeling. So there's pretty much just 4 or 5 different types of models that you'd have to kind of be familiarized with. And an important thing is that these models can all like be overlaid or used on top of each other. So each model has different pros and cons. And if like you're looking for a protein's like backbone R groups, you could always like overlay 2 to kind of get both. Um, so the first kind of model is the ball and stick or wireframe, and this pretty much shows each atom and its covalent bonds with each other, and also shows the R group or the backbone and R groups. Um, but the con in this one is that it's hard to discern the backbone from the R group. So it's just very useful for showing bonds without the actual like individual atoms. And then you have the spaceful model, and this shows each atom and the physical size relative to the other atoms that it takes up. So this is good for showing the overall shape of a protein, especially on the surface, and that would be important for R group interactions with say like ligands. But the con for this one is that it's hard to see the backbone of the protein. And then you have the ribbon model. This shows the secondary structures such as the alpha helices and the beta plate sheets pretty well, but you can't see the R groups in this one. And then lastly, you have the backbone model, which just shows the polypeptide like chain. But it doesn't show the R groups or the the atoms or like how much shape they take up. So it's a very good like outline or skeleton of what a protein would look like.
Sorry, one sec. Just the main thing for the models, guys, just knowing what they look like and what they show, in case you were asked a question where maybe you were given a picture of a model and had to figure out like which one it matches with, or being able to figure out like if a scientist was interested in studying secondary structure in a protein, what would be a good model that they could use, and like using what you remember from class or maybe your cheat sheet, you'd look to your sheet and you'd say, oh, the ribbon model shows alpha helices and beta sheets. So this would probably be the best one to use when the scientist wants to look at secondary structure of a protein. So does that make sense for the kind of thinking that would be required for that kind of thing?
And here we have like different kinds of examples of the different kind of models. So you can see how the ball and stick model. You can see how each of the red circles would be an atom, and the gray bonds between them would be the covalent interactions. You can see how the ribbon model shows the different kinds. In this picture is the alpha helices. The spaceful model shows the atoms and how much size they take up, and then how the ribbon model just shows the overall like path or shape of the polypeptide. And I will go over KD and ligands. So protein ligand interactions. What is a ligand? It's anything that a protein or an enzyme binds to. And it does so through a binding site, which can sometimes be a binding pocket like you see with CMP, or it can also just be a flat surface somewhere along the protein. But the important thing is that whether it's a binding pocket or a binding site, the protein's going to have very specific amino acids that are going to have to be able to make non-covalent interactions with the protein or the ligand of interest. So like you can see how here in this example, CMP is going to be making specific bonds, such as like a hydrogen bond with a serine, electrostatic interactions with an arginine. And you want to think about how like if it wasn't a serine there, then it wouldn't be able to make that hydrogen bond, right? And so it's very important for there to be specific amino acids in the right orientation for those non-covalent bonds like we were talking about to happen. And this is also like it plays back into like why it's important to have the right R groups and understanding like why their chemistry can allow them to make like the certain kinds of bonds that they can. So like again, you can see how a hydrogen bond, there's an exposed OH, and that pairs perfectly with the OH on a serine. So say like if it was like another amino acid there, like say if it was a glutamic acid there instead of a serine, then you wouldn't be able to make that same kind of hydrogen bond. Um, and then we can talk about affinity. So affinity is the strength of an interaction between the ligand and its binding site. And that's defined mainly by the number of bonds between a protein and its ligand, and also the strength of those bonds. Um, and also with that, also like the type of bond and its angle. And again, like from weakest to strongest, you have van der Waals interactions, hydrogen bonds, ionic bonds, and then the covalent bonds. And then we have the dissociation constant, or KD. And KD is pretty much a way to measure the strength of a protein ligand interaction. So again, the lower the KD, the stronger the affinities, because KD is basically just a measure of how long it takes for a protein and ligand to dissociate or break apart. And you can calculate that by the equation here, like PL over C, where P is defined by your protein, L is your ligand, and then it's divided by C, the complex, or the amount of protein and ligands bound together. And the important thing to know for that is that when you have half a concentration of ligand, or the binding sites of the protein are filled by the ligand, or you have P equals C, then you have, and that pretty much defines the smaller KD, which means that that's when the protein and ligand binding interaction that is at its strongest, or highest affinity. And again, like there's an inverse relationship between the strength or higher the KD and then the weaker of the protein ligand affinity. Oh,
I wanted to say as well that like the KD and affinity are inverse because if something had a high KD, it'd be more likely to disassociate. There's something called an association constant as well, but I think doing it with KD makes it easier to see a protein and ligand. But if you have a high KD, then that would mean that protein and ligand, they're more likely to separate from each other. So that's why a low KD, a low dissociation constant, means that two things are binding together very well and would have a higher affinity. And when we said before, affinity is based on like types of bonds and stuff, is based off of types of bonds and like the angle of bonds. That's why, like, if you were to substitute like a protein with like different amino acids, you can affect the kind of bond that it would make because of different category of amino acids make different bonds, and therefore by doing that, you can change the affinity. So that's how those things kind of tie together. Okay, can people hear me in the back? Okay. Somebody said no. So. Okay, how about now? Thank you. Okay, so now I'm going to talk about protein trafficking. Okay, so main ideas for this is that in eukaryotic cells, like humans, proteins are made in the cytosol by ribosomes. You guys knew that from 151. So in the cytosol, you have ribosomes, and there are two types we talked about. You have the freely floating ribosomes, and then you have the ribosomes that are attached to the membrane of the ER, the rough ER. Proteins that go into the endomembrane system are synthesized by ribosomes that are connected or that are attached to the rough ER membrane. And those proteins can go through the endomembrane system, which are the membrane-bound organelles listed there. And proteins that are not going through the endomembrane system, so these are your mitochondrial proteins, nuclear proteins. These types of proteins are made by freely floating ribosomes in the cytosol. She doesn't go into detail about chloroplast proteins, so just ignore that. But yeah, so cytosol proteins, obviously, mitochondria and nuclear proteins. Any questions about that so far? And that's just visual representation of what I just said. So you have proteins being made by ribosomes that float freely, and then ribosomes that are attached to the rough ER membrane. So endomembrane system, it's just a group of membrane-bound organelles that package and transport proteins, and it's composed of ER, Golgi, lysosome, cell membrane, and any of the membrane-bound organelles. And this is also called the secretory pathway, although I don't think she mentions that, but that's just something to know. And an important thing on this slide I'm going to mention is that if a protein only has an ER localization sequence, so an ER import sequence, the default mechanism of the cell is that it will have that protein travel through until it reaches the end of the endomembrane system. So the cell membrane and heading out, so it would end up being secreted out of the cell, unless there's another signal sequence that's in there that is like a retention sequence to put that particular protein somewhere. But the default cell program is that if there is ER import sequence and nothing else, like no retention sequence anywhere, that protein is just going to go through the pathway. Any a question over there?
I think you don't need the ER because you need to go to the ER to get to the Golgi.
Yeah, I think only ER import, but I think the question would specify if it needed any other sequence, but at least from what I remember, it was just like the ER import. And then it just kind of goes through the endomembrane system. And I had a picture of that, but I didn't put it in here, but I'll put it in there at the end. But yeah, I think it just needs the ER import because that just starts its way through the endomembrane system, unless it has like a retention sequence. Does anybody have any questions on that so far? Another question? Yeah, so that's the next slide. So I'm gonna go through that. But yeah, so this point might be confusing on here, but essentially what I was trying to say is just like if the protein only had ER import and like no retention sequence, that it wouldn't stay in the ER, it would just kind of keep going through this endomembrane system until it goes out of the cell. But most of the time you won't see that. So in the questions it will specify if there's some other sequence in there. But I am going to get to like the differences. So how proteins are trafficked is done through signal sequences. A signal sequence is a stretch of amino acids embedded in primary structure. Signal sequences are not added after protein is made. That's an important distinction because it's not a post-translational modification. It's built into the sequence of amino acids in the protein. And anytime a protein gets localized to the ER, has to have an ER signal sequence, like the ER import sequence. And the reason for that is because the ER import sequence is recognized by SRP, and that's just like a molecule that will bind to the N terminus, which is where the ER sequence is, and bring that protein to the ER. And if the protein does not have the ER import sequence, then it will not leave the cytosol. If it had no ER import sequence and no other sequence, it would not leave the cytosol, so it would stay in the cytoplasm. And like I said before, any protein that ends up being secreted out of the cell has to go through the endomembrane system. And once a protein leaves the ER, they rarely come back out again. Does anyone have any questions on what I said so far? Okay, go ahead. So what that means is that when a protein gets imported into the ER, there's a mechanism in there I'm gonna get to. There's like pictures of it in the next coming slides. But when a protein gets imported into the ER, the signal sequence gets cut off. It's an N-terminus sequence. So the signal sequence gets cleaved by a special protein called signal peptidase. You don't have to know the name of the protein that cuts off the sequence, but just know that the sequence gets cut. So it's unable to like get out of the ER. So yeah, that's what I mean by they rarely come out again. Because once it's being sent to the ER, also ER proteins have a retention sequence, which is in the C-terminus. So when you cut the N-terminus, the rest of the sequence, the rest of the polypeptide is red, and the C-terminus with the retention means that the protein stays in the ER. So I'm going to get to that in future slides, but did that make sense? Did that make sense? Just want to make sure it made sense. Any other questions? Yeah, so I have, I have the pictures from the, when I did the week 3 review. Yeah, I have the pictures from the week 3 review of that process. But essentially, it's, I think I mentioned in others in the upcoming slides, but when a protein goes to the ER, while it's being synthesized by the ribosome, let me see. While it's being synthesized by the ribosome, which is attached to the ER membrane, at the same time that that protein is being made, it's in the process of being synthesized or created. So the N-terminus is made first, and because the signal sequence is red, because the polypeptide is red from N to C-terminus, as soon as the signal sequence is created, then the SRP molecule will recognize that and attach to the signal sequence and import and pull that protein into the ER, even though it hasn't finished being created, like it's still in the process of being translated. So it's being pulled into the ER at the same time as it's being made. And it gets pulled into the ER kind of like spaghetti. So in its unfolded, unraveled form. And then once the protein is finished being made and it's in the ER, the signal sequence gets cleaved. And then there are chaperone proteins that help it to refold into the correct 3D orientation. Did that make sense? Any other questions?
You said that on the primary structure. So I feel like on one of the slides, the is
That's right. So that's correct. So there's a difference between proteins that go to the ER and mitochondria and proteins that go to the nucleus. And I have the chart with similarities and differences, but his question was just about the differences between where the signal sequence is. A signal sequence on a nuclear protein is going to be on the exterior surface. If you guys remember nuclear import and export, proteins like important and exportin will bind to the signal sequence, which is located on the outside surface, and carry those proteins into in or out of the nucleus. And then for ER and mitochondrial proteins, the signal sequence is embedded in the primary structure. So I am going to get to that, but that's the differences between the two. But it's still something that's part of the protein itself, like while it's been made. It's not something that gets added after a protein is created. So it's always like within that protein itself. That makes sense. It's not like a post-translational modification where the protein is created and then it's just tacked on with a signal sequence. It's always like included in the protein as it's made. So does that make sense? Okay. So just more on signal sequences. Like I mentioned before, proteins will enter the organelle unfolded for mitochondrial and ER proteins. And then the exception to that is the nucleus, as you said. Proteins will enter the nucleus fully folded and fully made, which means that they just enter in their regular shape, like the tertiary structure. And that's because the nucleus has the nuclear pores that can squeeze in the proteins. And the process is highly regulated. Another thing I want to mention is that signal sequences can behave as a binding site for another protein. So like I was saying earlier, signal sequences for nuclear proteins and signal sequences for the nuclear proteins, the important or exportin can bind to the NLS or NES on the protein and carry it to the place it needs to go. And yeah, I think most of the things in this slide I already said. And another distinction between your mitochondrial ER proteins versus the nuclear proteins is that the protein will enter the ER in the mitochondria in the unfolded state. So it kind of enters it looking like spaghetti if you see the cartoon pictures I had before. And so it enters it while it's being translated, like towards at the same time it's being synthesized, that protein is already being pulled in through the ER. And that's just to do with the mechanism of SRP, recognizing that N-terminus immediately as it's made, because it's the first part of it that's made, and pulling it into the ER. Does that make sense? Can I get like a thumbs up? Any questions on anything I said so far? No? So this is just a summary chart. Some of these slides are similar if you guys have been to my review sessions in the past. I just gave it a new theme and just kind of rearranged some words, but we have just a summary of nucleus, ER, and mitochondria, similarities of them and differences of them. You're welcome to take a picture, but I am gonna send out the slides at the end of the review session. But yeah, so those are just all the summaries of everything I said. And signal sequences stay on the nuclear protein at all times, which makes sense because it's on the exterior surface. So important and exportin can bind to it and bring it where it needs to go. ER and mitochondria, signal sequence is cleaved after the protein has been pulled into the organelle. And then it's refolded by chaperone proteins for both of these. And signal sequences for ER and mitochondria are part of the primary structure of the protein. It's part of the sequence of amino acids. And then for the nucleus, the signal sequence is part of the folded protein, so that tertiary structure, because on the exterior. And yeah, so signal sequences in ER and mitochondria N-terminus, and then signal sequences for nucleus are on the outer surface. And that's just a summary of what I just said. Does anyone have any questions on that? No? It makes sense? So signal sequences, there are different kinds of signal sequences depending on what organelle it's gonna go to. You don't really need to know like the details of what types of amino acids are included in a signal sequence. Usually if there's a question about that, she will give you the sequence of amino acids. But just know like the general trend of ER import signal sequences is that they have hydrophobic amino acids in there. And if you were asked a question about it, then and you switched like a hydrophobic amino acid to a different amino acid, then you could make some inferences about like how that would affect protein localization, because changing like a certain string of amino acids can change like the whole mechanism. And yeah, so the ER import sequences are N-terminus. You have hydrophobic amino acids followed by interspersed charge R groups. So if she gave you like 5 amino acids that followed that type of chemistry, and she changed one to make it polar, and she asked you to come up with like an idea of what could happen with ER import, then like you might be able to make predictions on like maybe SRP wouldn't be able to recognize the N-terminus sequence anymore because it's changed or the chemistry is different, like stuff like that. Those kinds of problem solving questions. I mentioned ER retention sequence before. That's found in the C-terminus. And that sequence is very important to have because it tells the protein to stay in the ER. And it's important for ER proteins that live and work in the ER. So proteins that are supposed to be localized to the ER, they have to have 2 sequences. So it needs ER import, which tells it to go into the ER, and then ER retention sequence, which allows it to stay in the ER. Otherwise, it's gonna float through like the endomembrane system if it just doesn't have the sequences it needs. And import in the nucleus just has sequence on the exterior, same with export, but import and export use different signal sequences. So NLS is associated with the nuclear import, and NES is associated with nuclear export. And I put a note about cytosol because I think she asked the ICLIP question in class about whether a protein going to the cytosol would have a sequence, but it doesn't have one because that's where protein begins its life. So it's like born in the cytosol. That's the way I tend to think of it. So it wouldn't need to have to be localized there. So it doesn't have a signal sequence there. Does anyone have any questions about anything? Okay, now I'm going to talk about nuclear import and export. Hang on one sec. So nuclear import and export. Sorry, I didn't realize it was like turned the wrong way. So for nuclear import and export, you can't really talk about those things without talking about GEFs and GAPs. So GEFs and GAPs are proteins that regulate RAN during import and export. GAP stands for GTPase activating protein. You don't really have to know what GAP stands for. If you're talking about GAP, you can just say the abbreviation, which is GAP. So GTPase activating protein just means that RAN itself is an enzyme, and the type of enzyme that RAN is a GTPase. And what it does is it converts GTP to GDP. I know those sound the same, so it can be a little bit tricky, but you can add it to your cheat sheet, which I never got for my exam. But so yeah, so GTPase activating enzyme, that's what GAP does. And because RAN is an enzyme, it has its own ability. We call it an intrinsic ability that RAN itself can hydrolyze GTP into GDP. And essentially what GAP does, GAP is not an enzyme. GAP is just simply a protein. GAP will bind to RAN and will help to accelerate the process of GTP hydrolysis. And that's just turning GTP into GDP. Because RAN has the ability to do it on its own, but it does so at a very, very slow rate. So these processes happen in the cell where you need them to happen again and again, and enzymes are reusable. So to quicken the process, RAN uses the help of the GAP protein to help it do that. And so, yeah, so when GAP binds to RAN, to active RAN, so when GAP binds to RAN, and if RAN is attached to GTP at that point in the cycle, which I'm going to show a picture of, it will help to accelerate, speed up the hydrolysis of GTP and GDP. And yeah, so does that make sense, what I just said? Does anyone have any questions? A question? Yep,
So, GAP isn't an enzyme, it's just protein.
just a protein.
And
Yeah,
is GAP an enzyme?
GEF is not an enzyme either. These are just proteins. It's good to distinguish between what's an enzyme, what's a protein, just because that tends to be like the overarching theme of the class. So enzymes, so anytime you think of an enzyme catalyzes a reaction or like causes a reaction to take place. And so RAN would be an example of an enzyme because RAN has the power to hydrolyze GTP and GDP. And the GAP is just something that helps it to do it quicker. So it's not, the GAP is not the one doing the hydrolyzing, if that makes sense. So that's why GAP is not an enzyme. Same with the GAP. Any other questions about that? That was a very good point. So GAFs essentially kind of do the, I would say, the opposite of what a GAP does. So what GAF will do is GAF will bind to RAN in the cycle. I promise I have the picture, but I just figured I'd talk about GAFs and GAP first before showing the cycle. So GAF will bind to RAN and RAN at that point is inactive, so it's bound to RAN GDP. And when GAF, the protein, not enzyme, so when GAF binds to RAN, it'll cause a change in conformation. We use the word conformational shift a lot in this class when we talk about proteins. That just means that RAN undergoes like a shape change, like a shape shift. So GEF binding to RAN causes a change in shape such that RAN no longer has a good affinity for GDP. So what does that mean? That would mean that RAN, which is bound to the nucleotide GDP, like lets it go because it doesn't have a strong affinity to it. So it lets it just when RAN binds to GEF, it wants to release GDP. And so GDP will float away from RAN's binding pocket. And so when that occurs, because this is happening in the cell, there's just an abundance of GTP molecules floating around in the cell. And because at this point, the GEF binding to RAN has caused a change in shape, and change of shape can cause a change of function. So by RAN being a different shape, it can no longer fit GDP in that space. So it creates a situation for GTP to bind to the binding pocket of RAN. And so when GTP binds to RAN, that causes RAN to become active. And so when RAN is bound to GTP, through the help of the GEF, it's able to be in its on state or active state. So it's able to do its job with nuclear import and export, which I'm going to show on the next slide. Does anyone have any questions so far? I just thought I'd put a diagram of that just so you guys can see what I was talking about. But you have RAN, which is off, and then when RAN binds to GEF, causes a conformational shift or change in shape, so that's no longer able to bind to GDP. So GDP falls out of the binding pocket, and that leaves room for GTP to diffuse in and bind to RAN, and that causes RAN to turn on. And then after RAN GTP's done its job and the nuclear import or export needs to be, it will deactivate, and RAN can hydrolyze GTP into GDP, but it binds to, the gap will bind to RAN and help it to hydrolyze GTP into GDP in a faster way, so that's able to release the GTP molecule and undergo like a shape conformational shift. So whenever two proteins interact, there's usually like a conformational shift there. Any questions on what I just said? Okay. Okay, so now to talk about the, sorry, I realized I was holding it too close. Now to talk about the export and import cycles. I thought I had import first. Yeah, I skipped a slide. So nuclear import. So it's good to have an idea for these pathways of what happens normally in a pathway so that you're prepared to answer questions that have to do with what would happen if I made a change or if there was some sort of mutation in a protein. By the way, you will be provided with the nuclear import and export diagrams on the exam. So if you already added it to your cheat sheet, I'm sorry, but you do get the diagram on the exam. So just good to know because it's hard to remember it. So basically what happens with nuclear import, you have importin, which is the protein here, and importin is looking for the cargo protein, which is going to be delivered to the nucleus, and importin binds to the cargo protein by attaching to the NLS, the signal sequence, located on the exterior surface of that protein, which you see here. Then after that happens, importin will bring that protein into the nucleus. And then what happens next is that you have RAN, which is in its inactive state, bound to GDP. GEF is going to bind to RAN, cause it to undergo conformational shift such that there's no longer has good affinity for GDP. And so RAN doesn't bind to GDP anymore. And because there's an empty pocket in RAN, and RAN is in a new shape now, it can accommodate for RAN GTP. So GTP will diffuse into the binding pocket of RAN and cause RAN to be bound to GTP, which is great because you want RAN to be active. And so when RAN is active, here, it's able to bind to important. And when RAN binds important here, this is essentially what you want to happen for nuclear import, because the whole point of this cycle is to bring the protein from the cytosol into the nucleus. So RAN GTP binds to important, so it can bring it back out to the cytosol. And that makes sense if you think about it, because important only has an NLS sequence, so that's localization from cytosol to nucleus. And so it can't leave the nucleus on its own. So it needs help from RAN GTP because it doesn't have an NES. Important only has NLS. So important will bind to RAN GTP, and when that happens, important lets go of the cargo protein because it's supposed to be delivered to the nucleus. RAN GTP and important together, they leave the nucleus. They get out here in the cytosol, and then what happens next is that we need RAN to let go of important so that it can go back and bring more proteins into the nucleus. And so for that to happen, RAN will bind to RAN will bind to the gap protein, will bind to RAN GTP and cause a conformational shift in RAN, and that causes RAN to let go of GTP, or I'm sorry, no, I said that wrong. So gap will bind to RAN, and RAN can hydrolyze itself, GTP and a GDP, but the gap protein binding to RAN helps it to do that at a faster rate. So RAN will hydrolyze GTP and a GDP, and when RAN goes from being bound to GTP to RAN GDP, it has bad affinity for important. So that causes, RAN GDP can't be stuck to important. So important gets released from RAN. And then it goes back here and does it again. Did that make sense? Does anyone have any questions on that? No? Yeah, sorry, I messed up in my explanation there, but yeah. Question?
So, out of this, if RAN gap was non-functional, important would accumulate in the nucleus, also, and then if RAN gap was non-functional, would important wouldn't be able to let them.
So you're asking what would happen if GEF was broken, essentially, like if GEF had like a loss of function mutation. So if GEF, so the way I try to think of it is kind of like step by step wise. So if GEF had a loss of function mutation, then if GEF has a loss of function mutation, then RAN is not able to switch from RAN GDP to RAN GTP. So if there's no GEF, then there's no RAN GTP. If there's no RAN GTP, then there's no way for nowhere for important to, I'm trying to think this through at the same time as answering it, but it takes a minute. So if GEF is broken, then there's no RAN GTP. No RAN GTP, then that means that there's no RAN GTP bound to important. So that would most likely mean that you would have this part of the pathway, everything before the mutation would happen. So you have important bind to NLS, it would come, the important and cargo protein would enter the nucleus. But then you'd kind of be stuck here because you have to have RAN GTP bind to important so it can let go of the cargo protein. So it would just be held up in that area, I believe, the nucleus, right? Wait, did that make sense? And then I guess for gap protein, like if the gap protein was broken, then if there's like no gap protein here, then you have everything else would happen as normal. But then when you got to this point, it would be difficult for nuclear import to continue because even though like RAN can hydrolyze GTP and a GDP, it does it at a super slow rate. So it's super slow and almost like not noticeable in a way. And when you have these processes happening a lot in the cell, it's just gonna probably end up causing nuclear import to like stall at that point. So all of the, so at that point you just have like all the important would be stuck to RAN GTP and there would be no way to separate the two. So does that make sense?
And it would hydrolyze. But it would be really-
So I guess a way to rephrase that is like GEF is required for RAN to go from RAN GDP, which is the off state, to RAN GTP, the active state. And then RAN GTP is like, so the way I look at it is like, I feel like RAN is pretty much like the star of the show in there because without RAN GTP, you can't get important to come back out to the cytosol and have the process continue happening. So not being able to have RAN GTP would be like very detrimental to the cycle. So yeah, so RAN needs GEF to go from RAN GDP to RAN GTP. But for like the nuclear import in terms of gap, RAN can hydrolyze GTP into GDP by itself, but it does it at a super slow rate. And since we're thinking about human cells where these processes have to keep happening at a very quick rate because like the cycles continue to happen and they need to happen at like a fast enough rate, it would almost seem like because it ends up stalling, that process just would almost be like little to nothing, if that makes sense. So I think that's the way you should look at it. So it needs gap and needs gap to make this cycle work normally. So I hope that helps to clarify it a bit more. It needs both of those to make it work. So if gap was broken, that would be considered like abnormal. The cycle wouldn't work as normal. Any other questions on this? But yeah, those are all really good questions. Yep.
I prefer what you were saying. So if gas is working, you would say, let's say the rate would be slowing down, you would say it would be like
Not
as if
gap,
it's not working.
gap.
Yeah, sorry.
Okay, yeah, just making sure.
Yeah,
we say both start with G.
So you would just say it was basically not working, or you say the rate, like for a question.
So do you want to speak to?
Yeah, I
feel
can
like-
kind of.
I would,
So-
go ahead, you wanna go?
So you're asking about like gap, right?
Yeah.
If gap. So like if you think about like what's happening is that you have the GTP molecule, right? And you're gonna over time lose one of the phosphates, so then it'll be GDP. So that's gonna be like you're gonna be breaking that bond, right? So like that is like favorable and naturally it's gonna happen, but it's gonna happen slowly. So that's why like gap is there to speed that up. But even if it was broke, and if gap wasn't working, then that GTP to GDP would happen because you're just breaking a bond. But if you think about like earlier, someone else was asking, is Jeff needed for the opposite from GDP to GTP? Think about how in that case, you're adding a bond. Like that's not going to happen. That's an endergonic process. Like naturally, that wouldn't happen. So you would need to have Jeff in that scenario to like make that.
Like just imagine the cell has hundreds of like importance and hundreds of these things. 'Cause you guys are only looking at one diagram. But if this kept happening over time, that like Rand being able to hydrolyze GTP to GDP, it's not going to be fast enough to like fix that problem. So all of your important is going to be stuck to red GTP, like hundreds and hundreds of that. So that's going to put the brakes on the nuclear import. So I'd say just think of it that way. So that's what I mean. What I mean when I say it's almost like little tree hopping.
And like even if you like tie back to earlier, like why is REN like GTP like better for like bonding to the protein, right? Because it has more phosphates, so it has more bonds to make that would make it favorable. So like if you're thinking about like why is that all happening in the like sequence that it does, like that's also.
But you know what? It's always good to like for questions like that, if you included like your perspective of it, because knowing what you know about Rand being able to hydrolyze GTP, you could offer your perspective, and as long as it's like an accurate statement, like you're not just making up stuff in like when you're writing out the answer, then you do get like some credit for that, like being able to come up with your reasoning if it's like a different approach. But I think the way that Dr. Francis approaches that is that just the fact that GAP is not working, that's abnormal to the cycle itself, and that would just hold up all the importance would be stuck to RAND GTP, and that would just like hold the brakes on the cycle completely. That after a prolonged period of time, that cycle just it would just pump the brakes on nuclear import, and that like RAND being able to hydrolyze GTP and the GDP would be almost like unnoticeable process just because of all the backup of all those things. Does that make sense? Yeah. Okay, so that's nuclear import. So now nuclear export. They may look the same, but they are slightly different in the mechanism. So just gonna try and walk through quickly what goes on. So now because it's nuclear export, we're starting off in the nucleus. You have the protein exportin. And so sorry. The first thing that would happen is you'd have RAND becoming active, right? Like before. So you have GEF will bind to RAND, and RAND at that point is bound to GDP. And so when GEF touches RAND, GDP causes a conformational shift. That causes RAND to let go of GDP, then GTP diffuses into the binding pocket of RAND. That causes, and the RAND GTP is now active to do its job. So RAND GTP will bind to exportin. And this is different than nuclear import, right? Because exportin is able to carry the cargo protein by itself and the place it needs to go. But with this one, exportin requires the help of RAND GTP to be able to carry the cargo protein out of the nucleus. The exportin cannot just pick up this cargo protein and bring it out by itself. It has to be bound to RAND GTP. So that's why we have this triprotein complex will altogether exit the nucleus. And then because at this point we have exportin bound to the cargo protein, and we need to drop off this cargo protein outside of the, we need to drop off the cargo protein into the cytosol. So then you have GAP that will bind to RAND and cause it to speed up its hydrolysis of GTP. So RAND GTP becomes RAND GDP, and RAND GDP just has terrible affinity for the exportin. So RAND GTP, once RAND is bound to GDP, has poor affinity for exportin. RAND will let go of exportin, and then because exportin cannot hold on to the cargo protein by itself, exportin will release the cargo protein. And that makes sense because we want to bring the protein into the cytosol, so from nucleus to cytosol. And that's just nuclear export. And yeah, so like I said before, exportin is not able to grab the cargo protein alone. It needs the help of RAND GTP. And so that's why you see the triprotein complex exiting nucleus. And then once GAP binds to RAN, causes a conformational shift in RAN, so that's no longer able to bind to GTP. So then RAN is bound to GDP here, and RAN GDP can't bind to anything, so it just lets go of exportin. And since it lets go of exportin, and exportin can't bind to the cargo protein without RAN GTP, which is not there anymore, so then it just lets go. Okay. Did that make sense? Any questions?
All right, so gene expression and regulation in eukaryotes. All right, so gene expression regulation, so this is like the production of proteins, right? So just know that cells carefully regulate gene expression. That's why Laura talked about it so much. So just a brief review of the central dogma of biology. So in eukaryotes, transcription is in the nucleus, the translation is in the cytosol, and then in prokaryotes, it happens at the same time within the cytosol because prokaryotes don't have a nucleus.
What's that called? Like, isn't there like a name for like translation?
Yeah, co-translation. Or I know it has a name
You
too.
don't need to know the name, though. But yeah, I'm sure.
I
it has a
definitely, I
name.
know it. Yeah, it
I know
definitely
you're
has a
talking about,
name.
but
I
you don't
couldn't
need to
find
know
it
it.
earlier.
All right, so just going back to the central dogma. So DNA is read by RNA polymerase to make an mRNA transcript. Then a ribosome reads the mRNA to know what sequence of the amino acid should be strung together for translation. The protein is then going to fold into a specific conformation due to their amino acid sequence, as we talked earlier through their R groups. So this just kind of goes down through it. All right, so transcriptional regulation. So transcription factors. So there's two main types in eukaryotes. So we have general transcription factors, and from the name, they can bind to a lot of different promoters. And as you can see here, it says transcription factors regulate RNA polymerase binding to promoter DNA. And without transcription factors, RNA polymerase cannot bind to DNA to transcribe. So the second type of transcription factors we have in eukaryotes is regulatory transcription factors. So these are more gene specific. So, and with regulatory transcription factors, we can have positive and negative regulation. So positive will do is exactly what the name says, enhance transcription, which means we have no more RNA polymerase binding, which means we have more transcription and more gene expression. And then we also have negative regulation, which is just going to suppress transcription when bound to DNA. Any questions on that? No? Okay. All right, so another thing about eukaryotes is their chromatin. So chromatin is basically histone wrapped DNA. Packaging determines which genes can be expressed. There's two main types of chromatin. We have euchromatin, which is an open state. So RNA polymerase and transcription factors can bind to DNA and do their thing. The outcome is that the genes are going to be more easily accessed so they can be transcribed and then translated. And then we have heterochromatin, which is the closed state. So RNA polymerase transcription factors cannot bind or reach the DNA because it's too tightly packed around the histone proteins. And then the outcome of that is the genes are not going to be transcribed. Are we all good with that? All right. All right, so now we have RNA modification and processing in eukaryotes. So RNA has to be processed and made stable into mature mRNA in order to leave the nucleus. So there's two main types of processing that Laura talked about. The first one was the 5' guanosine cap, which is covalently added to the 5' end of the RNA molecule. It's going to protect the RNA by preventing the RNA molecules to be chewed by nucleases, which would ruin the strand. And then we also have the 3' poly-A tail, which is added to the 3' end of the mRNA molecule and protects the mRNA from being degraded. It also helps with export out of the nucleus. So those are the two modifications that Laura talked about. Any questions on that? All good? All right, guys. So now we have RNA processing. So RNA splicing. So as we know, there's non-coding regions in pre-mRNA. which are the introns in our coding regions, which are the exons. So the introns must be spliced out before translation. So like I said, introns are areas that are transcribed but are not translated. Exons are areas that are both transcribed and translated. So this is all done by the spliceosome, which I don't think Laura went into much detail about that. So we also have alternate splicing. So I think Laura just talked about exon skipping in this class. I know also like exons can move around. So if you have ABC, it can be ACB. But I don't think Laura talked about that much. Yeah, so just quick RNA splicing. Are we all good with that? Awesome. All right, so now we have mRNA life and other relation things. So cells can control this ability of their mRNA, so mRNA degradation control. So cells can control the overall mRNA degradation rates and degradation specific mRNAs. So later on, we'll get into siRNAs, which are a type of small non-protein coding RNA that can cause degradation of other RNAs, and that's like exogenous. mRNA localization, cells can control whether mRNA gets localized to new areas. So the P bodies with miRNAs, which we'll also talk about later. An mRNA's lifespan can be regulated, and a cell has the ability to determine how long its life will be. So cells can control the half-life of an mRNA. And then just translational controls, cells can also control, how often ribosomes are binding to the mRNA. So a protein can block or help ribosome bind to mRNA by either decreasing or increasing the affinity of the ribosome binding site. Any questions on that? All right, so post-translational control, so protein activity control. So allosteric, do you guys know what that is? Right, so I don't have like the best definition, but if we have like an enzyme, it's an area where another molecule can bind to the enzyme, but just not in its active site, which can change its activity of the enzyme. We also have post-translational modifications, alterations of protein function after it's been translated. So we can covalently add chemical groups, which can change the structure of protein and therefore the protein's function. So I think Laura talked about phosphorylation, acetylation, methylation, and ubiquitination. And also, nucleotide binding, GDP binding can change the behavior or activity of a particular protein. So, kinases activate or inactivate. Also protein degradation, the UPR pathway that we went through, which I think we'll talk about later. I don't think we went through that yet. And then ubiquitin mediated proteolysis, so just basically when a protein can get tagged with ubiquitin and be brought to the proteasome for degradation. Everyone good on that? So this goes over eukaryote gene expression and regulation. So these yellow boxes or these yellow tags are where they're regulating. So we have transcriptional control, RNA processing control, mRNA transport and localization control, translation control, mRNA degradation control, protein degradation control, and protein activity control, right? So those are the main ones for eukaryotes. So I have another slide on here that compares and contrasts, but as you can see with prokaryotic cells, they don't have histone protein. So prokaryotes do something with their DNA called supercoiling, which is where they can take, like the plasmid DNA and wind it really tight. So I'll get into the other slide when we compare them. All right, so let's take a look at the similarities first, I guess. So eukaryotes and prokaryotes both regulate mRNA degradation rates, regulate mRNA localization, regulate rate of translation, and regulate protein activity and degradation. Now their differences are eukaryotes mRNA is monocistronic, meaning that one gene is equal to one protein. Eukaryotes also package their DNA into chromatin. They also have a nucleus, so mRNA processing and transport. And also know that, in eukaryotes, basal transcription factors are the main type of transcription factors. And in prokaryotes, mRNA is polycistronic, so one gene can equal multiple proteins. They don't package their DNA into chromatin. Remember, they use the supercoiling. They have no nucleus, so no nuclear export. And they also use a transcription factor called sigma factors. I think Laura talked about sigma 70. Any questions so far on this? All right. Oh yeah, so sigma 70. So this is the transcription factor that is in prokaryotic cells. So proteins that help bind RNA polymerase to the promoter. So sigma 70 binds to the -35 box and the -10 box in the promoter. So that's downstream of the transcription start site. It initially unwind takes place at the -10 box because there's more A and T base pairs. So do you guys know why A and T are the reason that sigma 70 is going to bind there?
Easier because two bonds between them.
Yeah, exactly. So there's only 2 hydrogen bonds between A and T, where G and C, it's 3 hydrogen bonds. So sigma 70 is sequence specific. So I think Laura also showed in her slides is how they basically just sequence a bunch of DNA binding sites and they kind of compare them. So that's why we have the Tata box here, which is one if you take genetics, that's comes up a lot. So it says here also why sigma is in sequence. It's while sigma is sequence specific, it regulates a lot of genes. It can bind to various sequences, so affinity changes based on the sequence. Any questions on sigma 70?
One thing I wanted to add is that there's a lot of different examples of gene regulation or regulation of gene expression on there. So it can get kind of complicated to remember those all. So maybe if I were you, I would possibly write examples of those on the cheat sheet if you have room, or if you can like write it really small on there, just so you guys have not that the other one.
This
No,
one,
that one.
this
Just
one.
like all the things that David just spoke about, the things, the different
That's
things,
it.
and like the different examples of like what would work as like RNA modification and processing, like RNA splicing, RNA modification and processing, like how does how does RNA get protected when it gets transported out of the nucleus, things of that nature. So being able to write examples of those, I think would be a good idea, just so that you can just look at your sheet and remember. You don't have to like memorize all those things. But I think that would be a good idea, something to write. Yeah, I just thought I'd mention it.
All right, so we're just going to end off with the trip operon. So overview of like what defines an operon or what an operon is. It's pretty much just a general term for any part on a DNA sequence that can be used to code for, say, like a gene or an mRNA, anything that can like any part of the DNA that can code for a product, such as like a protein or something. And so because of that, you're gonna have operons that all share like certain sequences amongst each other. So you're gonna have first and foremost, like if you see on this graph, you're gonna have the promoter, which is gonna stand upstream from all the other sequences. And that's gonna be important because the promoter is where the RNA polymerase is gonna bind to start transcription. So if you think about how the sequence after the operator, that's gonna be where if there is a repressor protein, that's where the operator is going to bind to. And you wanna think about like, say if during the exam you forget which is which, just think about how P is always going to be upstream. So everything's going to be everything to the right of that's going to be downstream. So if RNA polymerase does bind to a promoter and it's going down the sequence, if a repressor is there, it makes sense for it to be the operator because that's going to block it from the rest of the leader sequence and the other structural genes. And then you have the 2 types of operons, which are either inducible or repressible. And pretty much just like in the name, inducible operons are operons that can be turned on. So usually their default state is off, but they can be turned on in the presence of a specific molecule. And those are usually going to be genes that aren't that like important or aren't that vital to the cell survival. And on the opposite of that, you're going to have repressible genes. These are genes that you can repress or you can turn off because they're usually going to be on. And that's because their genes are going to be pretty vital to the cell's survival. For example, such as the trip operon or the tryptophan operon. So for the trip operon specifically, this is the operon that's going to encode for a protein that's going to make the trip or tryptophan amino acid, which is a very like essential amino acid, like regardless if you're a bacteria or like eukaryote cell. It's going to be an important part of a lot of other proteins. So pretty much we're gonna look at the trip operon in bacterial cells. And I think like what like really confuses people like when we go through the different like scenarios of what happens if there's low trip, high trip, what happens if there's trip continuation, is that you're gonna want to know that trip or tryptophan is playing three main roles in the operon. One is that it's gonna be like the main like end product, right? So it's gonna be what the gene's gonna encode for the amino acid. That's the first role it plays. The second role it plays is that trip's also gonna be a co-repressor. So if trip is available in the cell, it's going to bind to a repressor that's going to stop the production of that operon. So that's the second role that it plays, right? So you have it as the end product and you also have it as a co-repressor. But it also has a third role, which is it's going to make up part of the tRNA, right? Which we'll talk about later when we talk about how this operon has both transcription and translation. So let's go over like the scenario. So if there's low trip levels and the bacteria doesn't have many tryptophan and it's going to want to make its own. So then it's going to turn on the gene product, right? There's no tryptophan to act as a co-repressor. The trip operon is repressible, so it's usually always going to, the default is that's going to be on. If there's high trip levels, then the bacteria has an abundant access to tryptophan. It doesn't need to waste the energy to make its own. So it's going to use its own like trip amino acids to act as a co-repressor, right? It's going to bind to the repressor molecule, and that's going to go on top of the operator part of the sequence. And that's going to pretty much block, again, the RNA polymerase from coming down the sequence. And then, like, if you want to think about, is a trip repressor a positive or negative regulator of the operon? You want to think it's a negative regulator because, again, it's turning the whole sequence off. And here again is like a diagram that shows the whole operon. Again, like you want to think about the sequences going, it's all going from the P, the promoter, all the way to the right. So everything to the right of the promoter is going to be downstream. Promoter is where the RNA polymerase is going to bind to. The one after that, the operator is where the repressor is going to bind to. And then you have the leader sequence, which we're going to talk about later in attenuation. And then you have trip E through A, which are just like structural genes that on the mRNA sequence, when it's transcribed, that's what's going to make the actual amino acids for the protein of the protein that's going to make tryptophan. So is that part clear before we go like farther? Oh, yeah.
And is the operator technically a part of the promoter?
I would think of it as its like own region. There's just those are just the two regions that are going to start the, so like everything after the leader sequence, the structural genes, those are genes that are specific to this like operator. But the promoter operator, that's going to be, those are two parts in like many operons. So I would think of them as like two distinct things, but they're like the like generic pieces of like any operon is going to have a promoter and then an operator sequence. And then leader sequence, I don't know if you'd have to know specific, like in this it would just be like the different attenuator regions, right? Like the 1, 3, and 4. I don't think you'd have to know like specifics of like if they're like called or anything, or if they have like yeah. And they're only numbered because of the fact that later we'll talk about how they're complementary, so they're kind of like, you know, form hairpin loops with each other. Anything else? So we have scenario 1 of 3. What if there's high levels of tryptophan available to the cell? So because of that, like we've been saying, tryptophan is the co-repressor in this operon. So if there's a lot of it, then it's going to bind to the repressor protein. That's going to activate the repressor and it's going to bind to the operator. And if it's bound to the operator, then well, the RNA polymerase, it can, then it's blocked from binding to the promoter. And there's going to be no transcription. That's going to start. So that's probably like the most like straightforward one of the 3 scenarios. If there's a lot of tryptophan, then it's not going to bother transcribing its own. And then we have an overview of attenuation. And pretty much attenuation, just what it means is any early like termination, in this case, like of specifically transcription of the trip operon. So pretty much like we were saying earlier with the leader sequences, um, in this case, the attenuation sequences 1, 3, and 4, they all are coded or need to be coded for the trip proteins. Um, and like we talked about in class, there's going to be 4 regions, and where there's like 3 regions of complementary. So like if you want to think about what's happening here is that RNA polymerase is coming in, it's transcribing from the DNA, right? It's producing a long mRNA chain. And like as you know, mRNA is just like one strand, so like the nucleotides are exposed. And if they happen to be complementary to each other, then that mRNA strand's gonna like form, like close in and like make loops on itself. Like we were talking earlier about how like non-covalent interactions are spontaneous. That's just by its nature of being RNA that complements each other. It's gonna like, you know, close in and make these loops. And these loops here can either happen between regions 1 and 2, regions 2 and 3, or regions 3 and 4. And later we're going to talk about the different scenarios of like when those hairpin loops are going to happen. And again, like this can happen because in prokaryotes, transcription translation's happening at the same time. So you want to think about, on the one hand, RNA polymerase is coming in, it's producing the mRNA, right? But then also right behind it is going to be a ribosome that's going to attach the mRNA as it's coming out, and it's going to then wait for the tRNA tryptophan to like then bring on like, you know, the anticodons and everything. And then that one piece is important, that the ribosome has to wait for the trip. But we'll talk about more of that later. So then you have scenario 2, like what if there's low or almost no tryptophan available to the cell? Then in that case, there's nothing for the repressor to bind to, so it's going to be inactive. And RNA polymerase can, it's free to bind to the promoter and start transcription. And it's going to produce no attenuation, like there's no problem. It's just going to fully go through all the transcription and translation. And the mechanism behind that, like we were talking about earlier with the hairpin loop. So let's see. So like we were saying earlier, as this is happening, regions 1 and 2 are going to be able to form together. They're going to make a hairpin loop. And that's important because this allows the RNA polymerase to stop, and this lets the ribosome catch up, because again, we talked about the ribosome has to wait for an available like tRNA, like tryptophan molecules to like physically come over and bring in like the anticodon regions to make the amino acid, right? So if it's able to pause at the first region, then that hairpin loop, it's going to break because ribosome can break the hairpin loops, and that's going to free up region 2 to bind with region 3. And that's going to allow the ribosome to then like, you know, catch up with the RNA polymerase. And then it's going to break the 2 and 3 loop, and then it's going to be able to fully make the entire transcription and translation. And what you're going to get from that, you're going to get the full mRNA sequence. Does that make any sense? Does that make sense? Like how first you're going to have the 1-2 loop, it's going to pause there, the ribosome is going to break that loop, and because that's broken, then the 2 and 3 regions can make a loop, and then you can like go on and then make the whole. Versus scenario 3, like what if there's low to medium levels of tryptophan, right? So in this case, like what we were talking about earlier, tryptophan is playing 2 roles in this scenario. One is that it's going to be the co-repressor, but it's also going to be the one that's physically bringing the anticodons to the ribosome molecule. So in cases where there's medium levels of tryptophan, right? There's not enough tryptophan to bind to the repressor molecule. So initially the transcription is going to start, but there's not enough tryptophan to act as a tRNA to bring to the ribosome. So what happens is that RNA polymerase starts, it binds to the promoter and starts transcription, but then later on the cell realizes that there is enough tRNA present. So what you have is attenuation, which is the premature stop of transcription, resulting in an incomplete mRNA strand, which can't be used to make any like tryptophan molecule, and thus like there's not going to be any tryptophan. So the mechanism for this, like before, there's going to be that 1-2 pause at the region 1-2, that hairpin. But in this case, there's enough TRP for there's enough TRP to bring enough anticodons to the ribosome, so it's not going to have that pause. It's just going to keep going along. So if it doesn't have that pause, there's not going to be a breakage of the 1-2 hairpin. And because of that, then regions 3 and 4 are going to make a hairpin loop. And because if you want to think about it, like what's physically happening, now you have 2 hairpins, which are going to like physically, like, one's going to fold in on itself. So it's going to yank the rest of the mRNA out of the mRNA polymerase, which is going to have an early termination of the entire process, and it's going to stop transcription translation. Does that make sense? How, because there's in this scenario 2 hairpins happening, you're physically pulling the mRNA out of the polymerase, and that's going to prematurely stop the whole process. Does that make sense? How? Yeah. So yeah, a summary of the 3 scenarios. 1, the simplest, if there's a lot of tryptophan available, then there's no problem. The cell doesn't need to waste time making any of its own. If there's low levels of tryptophan, then there's no repressor and transcription is going to happen normally as it should. You're going to have a 1-2 hairpin loop form. RNA is going to, or ribosome is going to pause, which is going to allow for that hairpin to break, which is going to free up region 2 to make a hairpin with 3. So you're just going to have the 1, 2, 3 hairpin, which is going to allow for full transcription because there's ribosomes can have enough time to catch up with RNA polymerase. And then in situation 3, if you have medium levels or low to high levels of tryptophan, you're going to have not enough tryptophan to act as a co-repressor, but you're going to have enough to act as a tRNA. So because of that, there's no chance for a ribosome to have that pause at region 1. And if that happens, then 1 and 2 are going to make a hairpin loop, and 3 and 4 are going to make a hairpin loop. And because you have 2 loops, right, you're like, think of like if you're coiling a rope, then it's going to get shorter, right? So because of that second loop, you're going to physically pull that mRNA out of the polymerase, and that's going to stop transcription translation. And you're going to have an attenuated or non-functional mRNA and then no protein. So is that, oh yeah, and then we have
Okay, so this is the last category for exam-related stuff today. You guys have been awesome. Thanks for hanging in there. This is the last thing. I kind of panicked while I was sitting here because I realized I forgot to mention something before with signal sequences. Something really important you guys should know is that based off of like all the signal sequence organelles, like the ER import sequence is the one that happens the quickest, I'd say, because with ER import, you have, as soon as that protein is synthesized, like the N terminus, as soon as that little part of it is made, SRP will bind to that and pull the protein into the ER. And that's a very quick process compared to any other localization process. So that is one of the fastest ones. I just figured I'd bring that up just in case and that you guys have that information. Yep.
If the mitochondrial one is also in the end, does that mean that that one's also as fast, or is it something like the other one?
No, this is just the SRP and the N terminus sequence and that process of bringing it into the ER. That's something specific to ER proteins. For the mitochondria, the mitochondrial transport diffuses into the way that mitochondrial proteins go into that. I don't think she went into much detail when I took the class, but it has like a different process, but it doesn't involve SRP. The SRP, the N terminus, and the protein being pulled into the ER lumen, that's done, that speedy process is done with just ER localization. And I think with the mitochondria, the protein is synthesized and then it goes through the mitochondrial membrane, I believe. I'll have to look into that and see if I can email you guys about that. You have a question?
So in class we talked about how So like the ER, it's like it had. But what if it was like a mitochondrial signaling sequence and a
So if you had mitochondrial and nuclear, I don't think she would ask something like that. I think one of the main focuses, the main organelles I think she focuses on is nucleus, nuclear import, ER, and nuclear import mitochondria, but she wouldn't, I think if she was to give you some sort of question like that, based on like what you've learned about the class, she could throw in potentially like if I had a protein that had like ER import, maybe like ER import and like maybe something random, some like mitochondrial retention or something, and nuclear localization sequence, like where would the protein end up? And just it would be based on like a timing thing. So just because like you what you know about signal sequences is that ER import is N terminus, so that sequence gets read first. And so if the protein, if there was a like a made up protein that had like ER import, ER retention, nuclear import, the first thing that gets read is kind of like a race to get to where it needs to go. The first thing that gets read, because in the N terminus, read N to see the polypeptide would be the ER import. And then at that point, as soon as that little part of the protein is made, just the N terminus, SRP's already grabbed it and pulled it into the ER. So by the time the rest of the sequence is read, that nuclear localization sequence that's on the exterior surface wouldn't even matter at that point, because at that point, once the protein's fully synthesized, we know based on what we know about ER import and the way it works, the mechanism, it's already in the ER and it's gonna be retained in the ER if it had an ER retention sequence. Yeah, so I just thought I would clarify that. And then if a protein only had like a retention sequence and nothing else, then that retention sequence is useless. So if a protein just had like ER retention sequence and nothing else, the protein would just stay in the cytosol. And that's because you need an ER import sequence to get it to go to the ER first, and then the retention sequence. So ER localization, you need two sequences to make it work. If a protein is going to live and work in the ER or stay in the ER, it needs ER import and ER retention. Does that make sense? And the ER import is N terminus, and ER retention sequence is C terminus. And so that's why even after the protein makes its way into the ER, the N-terminal sequence is cleaved, the ER import cleaved, and then it stays in the ER because it has that ER retention sequence. Does that make sense? Okay, so sorry to like backtrack, but I just thought I would mention those things just to clarify. Okay, so now we're actually gonna go to the last concept we have, which is RNAi. So main ideas here is that RNAi either results in mRNA or RNA degradation or the suppression of translation. And so a key factor in that is you have the RISC, which is the RNA-induced silencing complex. You don't have to know what it stands for. If you were to ask to talk about it, you can just call it RISC, but I just figured it'd be interesting to put what it stands for. RISC is RISC has two parts to it. So it has RISC and then it has something called argonaut. Sometimes you'll see it in diagrams abbreviated as like AGO, but the argonaut is the enzymatic component of the RISC complex. So that's what ends up like destroying mRNA and the mRNA degradation. And then RISC is just the protein that's part of the RISC complex. And RISC, what it does is it binds to a small guide RNA and uses the guide RNA to target other RNAs using something called base pairing complementarity. There's two types of RNAis we looked at. I didn't say it before, but RNAi stands for RNA interference. So they're just like different kinds of RNAs. The one that we have in our genome for humans, which I just want to emphasize, the one we have for humans is microRNA. I think sometimes it's abbreviated as miRNA. And then a kind that comes from outside that has to be injected into cells and stuff like that, that's siRNA, so small interfering RNAs. So those are from exogenous sources like viruses. And most of the time, those siRNAs, like the pathway leads to mRNA degradation. And then for microRNAs in human cells, the pathway can lead to translational repression, but sometimes you can get mRNA degradation. So now I'm just going to talk about the two the two different types. So mRNA, the one that humans have, I said it a third time, so just make sure that it sticks in your head because that question may or may not appear on your exam. Also, this diagram is not in the exam. So cheat sheet, if I don't know if you guys already created the cheat sheet, but maybe start over at this one and add these ones because this diagram I know for a fact is not in the exam. So having notes about it or understanding what's going on there is a good idea. Okay. So miRNA, a way to remember it is I just think of it as like my RNA, like miRNA. That's how I remember that it's in my genome. So it's endogenous. So the one human use. I'm gonna say it again. So basically what happens here with the miRNA pathway, the RNA, when it is in the, I think the way I'll describe it is I'll have with the picture and just do the words here. So when the RNA is initially transcribed in the nucleus, it's called pre-RNA, pre-mRNA, which it doesn't have in this picture actually. I'm gonna correct that in the picture. But yeah, so when it's initially made, it's called pre-mRNA. I'm just gonna leave the word, just describe it that way. And then in the nucleus, you have drosha, which processes and cleaves the pre-mRNA into tinier pieces, into smaller pieces, called pre-mRNA. Yeah, so I'm confusing myself. So there's PRI mRNA, which is what it starts with. And then in the nucleus, drosha processes and cleaves PRI mRNA into smaller pieces called PRE mRNA, which is pre-mRNA. They're both pronounced the same way. So I'm just emphasizing that they're different though. They're different. That's why the colors are different. And so pre-mRNA, the PRE one, exits the nucleus. And in our diagram here, we have exportin that helps it to do that. But yeah, so at that point, pre-mRNA exits the nucleus. And then once it gets into the cytoplasm, you have dicer, which will process and chop up the pre-mRNA into even tinier pieces. And these pieces are called mRNA duplexes. They're about 20 to 25 base pairs. And at this point, your mRNA is double-stranded. So then the important part of the pathway, we have the RISC complex here, and it will come in and it will bind to the miRNA. So the same 20 to 25 base pairs in step 4. And RISC, what it will do is the RISC complex will unwind 2 strands, and it will take 1 strand to use as the guide strand. And it will take us, then that takes a single strand of RNA and uses 1 as a guide strand and discards the other strand. So it just gets rid of the other strand. So 1 strand is used as a guide strand, so it will stay attached to RISC. And the other one just gets degraded. And the one that gets degraded is called passenger strand. But I don't know if she goes into detail about that, but just know that 1 strand is a guide strand. The one that isn't chosen is discarded, and that one's called the passenger strand. So then RISC with its guide strand is able to go around and target mRNAs and either cause degradation or translational repression, depending on the mRNAs it binds to. And so when you have the RISC with the guide strand, it's going to use base pairing complementarity to target the RNAs. And so if you have perfect target complementarity, this will lead to mRNA degradation. And the degradation is performed by the Argonaute part of the RISC complex. It's just the enzymatic component of the RISC complex. And then if you have the opposite of that, you have the imperfect partial or like partial perfect binding that leads to the RNA being stored in the P bodies. And most commonly in humans, that is ends up leading to translational repression. So that's why I have the note there. In humans, it's typical for mRNA to end up being silenced rather than degraded. So the mRNAs that get that are targeted by the risk and guide strand get taken to the P-bodies for storage. But sometimes when they're in the P-body, they can get degraded as well. So that's just what the image is showing there, like the end result. So imperfect target binding, translational repression, or perfect target binding and mRNA degradation. Yeah, so does anyone have questions on that pathway so far? No? Okay, and then the other one is siRNA. This one's not used by humans. It's the exogenous source one. So you'd have a virus that would be injected into a cell. So what happens here is that it starts off in the cytoplasm. You can have double-stranded RNA, or I think shRNA is short hairpin RNA, and that gets cleaved by dicer. So similar process to miRNA. Everything that's happening in the cytoplasm is similar here. So the exact same thing as the miRNA, except you're just starting from like the cytoplasm onwards. So the same proteins at play here. So you have a dicer that cleaves the double-stranded RNA into smaller pieces. Then you get the miniature siRNA duplexes. And then the same thing will happen. The RISC complex will bind to the siRNA duplex, unwind it, pick one to be the guide strand, and then the other one will get discarded. And yeah, so pretty much the same idea here. There's the complementary base pairing. Then if you have an exact match with the base pairing, you get cleavage of the miRNA or the mRNA degradation. And yeah, you have a question?
So when it goes into the cytoplasm, it starts off as dsRNA or shRNA. Then like what do those stand for?
I don't know. That's just the image I got from Google,
I know.
but I think either one.
I think DS just stands for double strand.
Double-stranded RNA. But I think it's just saying that like you could have the double-stranded RNA, or you could have like short hairpin RNA. I think it's just saying that when you inject the virus in there, you could have either. Yeah, it's a picture I got from Google, which I know it can
But it just doesn't
just make it
become-
misleading.
It doesn't become known as siRNA until it's duplex. And it gets processed.
That's correct.
Okay.
Yes, sorry, I don't mean to confuse you guys, but yeah, so just ignore the shRNA and just go from dsRNA and do that. But it's just, I think the image is just trying to say that you could either start with shRNA or double-stranded RNA. I think that's what it's trying to say. I can ask Tina as well, because these are her slides from week 4. So yeah, any more questions? That was a good clarification question. Can you say it one more time? And hang on one sec.
We kind of talked about this in like a help session with Laura last
Mhm.
week. That basically just it can be reused. I don't know if that's like. what
I think
it's
that's
saying now,
that's true.
But yeah, it's like not a 1-to-1 ratio of like the siRNA to like the RNA.
Yeah. Yeah, I think that's the answer. I wasn't sure, so I was trying to like look at my notes here to figure it out. But I think that is the correct answer. No, it's not a 1-to-1 ratio. But I don't think she goes into much deets on that in the exam. The main takeaway point from these is just establishing what goes on in both pathways, and just remembering that for humans, like typically you see translational repression happening, the miRNA pathway, and siRNA more likely for mRNA degradation. But just similarities and differences between the pathways is good to know for these. Another thing I thought would be interesting to mention is we talked about the mechanism of how RISC just unwinds double-stranded RNA and picks one to be the guide strand and throws the other one away. So an interesting question that sometimes I get asked is, why doesn't RISC just grab a single strand of RNA to begin with, if it only is going to use one. So the reason for this is because in a cell, cell's a very hostile environment. So single-stranded RNA tends to be more unstable than double-stranded RNA. And what we mean by that is just it's more susceptible to be destroyed. The cell nucleases just kind of go around and start degrading things in the cell. And so if it has a single-stranded piece of RNA, it's just more likely to get degraded by a cell nuclease compared to double-stranded RNA. So just think of it as like a single-stranded RNA is just at risk of being like eaten by a cell nuclease. So yeah, and so as a result of that, if you have a single strand of RNA just lingering in the cell alone, it just gets to, it can be degraded so quickly that it wouldn't, risk would not be able to find it or bind to it. So those reasonings show that the process of risk being able to select the guide strand, like to be able to pull apart or unwind a double strand RNA and select the guide strand and discard the other one, is very vital to the RNAi pathways. So the way that risk normally operates is that it just has to unwind and pick one and discard the other one. So that would mean that RNAi most likely wouldn't work if you had just single-stranded RNA and if you try to throw it at risk and see what would happen. So that proves that risk needs double-stranded RNA to do its job. Any questions about that?
Oh,
Yeah.
Like bound to the list for a long time with like to create other RNA. Does that happen with mRNA?
Can you say it one more time?
Yeah. So for siRNA, that one single strand gets bound to the RISC complex, and that's why it can degrade other mRNA because it's bound to that. Does that happen to the mRNA?
Actually not sure.
Like being bound to it.
Mind saying it worked like that last part of it one time? Or your last part of your question?
Yeah, so the siRNA, the single strand, is bound to the RISC, which then leads to degrading mRNA. So does that happen to mRNA? Just like being bound to it and then being able to degrade other travelers like the one.
yeah.
No, so both of these have the ability. So in both of them, they both involve risk, and risk in both of these will bind to a double-stranded, smaller piece of RNA, whether it's siRNA, mRNA, unwinds the strands, chooses one to be the guide strand, and the risk complex and that guide strand will go and target other RNAs in the cell and either cause mRNA degradation or translational suppression. That can happen in both. We were just saying that for human cells, the mRNA pathway, it's more common for when we look at human cells that you have translational repression happening there rather than like the human cells just destroying all the mRNA. But mRNA degradation can happen for those. So that brings me to similarities and differences between mRNA and siRNA. So yeah, they have similar things involved, but basically the way I look at it is siRNA pathway is basically follows the same steps as mRNA after it makes it to the cytosol. So it's just the siRNA pathway is like the mRNA pathway from the cytoplasm onwards. If that makes sense. So similar steps. Any questions?
I have a question. So like we were talking about how like siRNA can like be reproduced multiple times. Can mRNA be reproduced multiple times? Like
I'm not sure. That's more of like a hypothetical, which I don't think she'll ask you. So yeah, I'm not too sure on that.
I know like siRNA can be used more often, so it doesn't have to be a one-to-one base pairing. Like mRNA is
Oh, I think MR.
mRNA, I think mRNA is 1-to-1 base pairing, right?
Oh, no, that's like, no, you're.
So mRNA or MIR, I'm sorry, I think mRNA. miRNA is like the 1-to-1 base pairing, so it's used. I feel like it's used multiple times. I feel like I've heard that before. But it's used, I guess, it has less situations where it's used compared to siRNA, where siRNA doesn't have to have 1-to-1 base pairing for every mRNA that it's either, I don't know if it's 1-to-1 where it degrades it and the other one is.
I think it's I think the way it is that mRNA doesn't have to be an exact match. So like a bunch of different, since it doesn't have like one specific target, it can be used for multiple different ones. And those are all targeted for not, since it's not a complete match, so none of them are targeted for degradation, they're targeted for sequestration, which is useful in eukaryote cells because there's so much more going on. It makes more sense to just sequester something rather than just degrade it, and you can use it again. But then in siRNA, it is like an exact match. So there's only one thing you can use, and because there's only one, it makes more sense to just degrade it rather than just hold on.
Yeah,
Does that make
those,
sense?
sorry, those like little details of it, like I don't think she wants you to focus on too much. It's just the main idea is like what happens in mRNA pathway, what happens in siRNA pathway, and like the importance of like the roles of the proteins in there, particularly risk complex and stuff like that nature. It's not going to be that deep. Like, does this one have a 1-to-1 ratio exactly? Because I don't remember her going into detail about that when I took the class, so don't think so. Just the overarching ideas, the main ideas behind these two pathways. Any other questions? No? Well. So thanks for coming to the review session. Thanks for sticking with us. It was a long review session. Thanks for all your questions and good luck with studying for the exam.